Hello everyone, my name is Egbert Einstein, biologist extraordinaire. I know you've seen this over and over, get it, over, by now, but I'm here to talk to you about the lac Operon. Now that I've used all my puns, let's get cracking, shall we? The lac Operon is a region of DNA that has a specific function, that is, the production of lactose digesting enzymes. The specific function of the operon means that it also possesses a very specific structure. The operon is expressed by a protein called RNA polymerase. The operon consists of a promoter region, which is where transcription of the genes begins. Think of it as the front door to the operon. A repressor gene responsible for producing proteins that stop the operon from working, the key to the front door. An operator site, an area which acts as the lock to the door. This is where the repressor binds to the operon. Structural genes, which contain instructions for the formation of digestive enzymes. These are the reason the lac operon even exists.
Now, just like the way you don't always leave your front door open, the operon is not always at work. A protein called the lactose repressor can activate or deactivate the operon, depending on whether it's needed. This is determined by whether or not the cell needs to digest lactose. In short, there's no point in the operon being expressed and producing lactose-digesting enzymes if there's no lactose to digest. Let's look at how, when lactose is absent, the LAC repressor binds to the operon, meaning RNA polymerase cannot bind. This is the repressor protein, and beside it is the sugar lactose. When lactose is present, it changes the shape of the repressor, and like a bent key in a lock, the repressor becomes useless. Now that the repressor is out of the way, RNA polymerase is free to bind to the operon and do its thing. Expression of the operon can now go ahead. But that's not all. Another system exists controlling the expression of the operon. Now then, the CAP protein helps improve expression of the LAC operon and is regulated by the cyclic AMP, which is in turn managed by the amount of glucose present in the cell. As the cell only wants to digest lactose if there's no glucose available. If glucose levels in the cell are low, then the level of cyclic AMP will be high. Just how cyclic AMP is involved is as follows. In low glucose levels, the cyclic AMP can bind to the cap, changing its shape and allowing it in turn to bind to the operon. With the cap protein and RNA polymerase bound to the operon, the expression of the genes is boosted up to 50 times higher than when the cap isn't present. This means that only a little lactose is digested when glucose is present, but lots can be digested when it's the only thing around. All this expression only ever results in one thing, literally. As the RNA polymerase moves along the operon, it creates one long strand of messenger RNA, which codes for the enzyme on that operon. And all of that work boils down to these. These enzymes work together to break down for use as food in the cell. I hope your brain isn't too scrambled by all of this. Now I'll be off now. I have a funeral to attend.